Hello, my name is Mariana Casas, and in the first place, I would like to thank the organizers of this symposium to give me the opportunity to talk to you today about our results. My group have been working in the last years in understanding the mechanism by which adult muscle fibers decipher the stimulation patterns coming from motor neurons to define their phenotype to adapt to its demands in a process called muscle plasticity. We will talk in the first place about our previous work. So we have shown that after 20 Hz electrical stimulation of adult muscle fibers, a process is triggered to activate transcription that starts with the activation of CAB 1.1. CAB 1.1 is then the trigger of this process, and we can see that when we use fibers pre-incubated with nifedipine to block CAB 1.1, and when we do that, we eliminate all the events that are downstream CAB 1.1. In the first place, release of ATP through panexin channels and IP3 production and change in gene transcription. So this ATP release that is produced through panexin channels induce also an increase in IP3 intracellular levels. These IP3 intracellular levels, in the same way that we have two peaks of ATP release, will happen also showing two peaks that are related to this ATP release. Importantly, all these events will end with an increase in mRNA levels of slow isoforms of troponin I and a decrease of the fast isoforms of this gene, a change that is a feature of muscle plasticity from fast to slow phenotype. We wanted to know if ATP could be a manner that cells use, a molecule that cells use to decipher the different patterns coming from motor neurons because this ATP release was frequency dependent. So for that, we stimulate the fibers with external ATP, 30 micromolar ATP, and we found that mRNA levels, changes of slow and fast isoform of troponin I were the same that we have already observed when we stimulate the fibers at 20 Hz electrical stimulation. Accordingly to that, addition of external ATP will induce an increase in IP3 intracellular levels, as we can see here, and that is in accord to the transcriptional events that we observe. So I show you this cartoon that summarizes the results I have shown until now. So in this cartoon, we have the T-tubule of our membrane, our muscle fibers that can be stimulated electrically at 20 Hz. This stimulation will be sensed by the voltage sensors of CAB 1.1 or the HPR and will communicate to panexin 1 to open it and allow the release of ATP to the extracellular medium. This ATP could activate its cholinergic receptors to activate a signaling cascade that will end with IP3 production, the binding of this AP3 to its receptors located in the nucleus and the sarcoplasmic reticulum mostly, to cause the opening of IP3 receptors that will allow a calcium signal that in turn will activate transcriptional change related to muscle plasticity from fast to slow phenotype. However, this cartoon is far from being complete, and a lot of questions still to be answered. So in this presentation, I will talk to you about 3 of these questions. In the first place, I want to talk to you about what happens to membrane potential after electrical stimulation or external ATP exposure. These questions come from previous work, principally from Andrew Voss' work, that shows that near the neuromuscular junction, there was an activation by ATP of cholinergic receptors, P2Y receptors. And this activation could be able to close some chloride conductance, allowing the hyper-excitability of the fibers. So in this context, and given that we also have release of ATP, we wanted to know if this ATP was able to change membrane potential. After, we will see what happens with these 2 peaks of ATP release and their roles. So the first experiment we made was take some fibers to measure the resting potential and measure the membrane potential after the addition of 100 micromolar of ATP. And we can see here that the muscle fibers depolarize around 20 millivolts with the stimulation of ATP. Another experiment we made was measure the membrane potential of the fibers over the time. And we made this experiment in presence or absence of ATP stimulation. So in black, we have the behavior of membrane potential of fibers that were not exposed to ATP. And in red, you have the behavior of membrane potential when ATP at 30 micromolar was added in this point. So you can also see that fibers are depolarized compared to control. Finally, we will ask us if the original stimulus that was the physiological stimulus that will happen probably in vivo, that is depolarization of the fiber by action potentials at 20 Hz, will also be able to depolarize the fibers. So we do the following experiment. We measure the basal resting membrane potential of an amount of fibers. We stimulate them with 20 Hz electrical stimulation, and we observed transient depolarization that occurs with an amount of depolarization also about 20 millivolts. Interestingly, this depolarization was recovered to normal resting potentials after 12 minutes of ending the stimulus. So at this point, we have that our pieces start to fit each other to form the puzzles. And we decided to make an experiment that will open a new way to see this process. So we decided to stimulate fibers with ATP, an experiment we have already done multiple times, and found the expected transcriptional change as you can see here. We decided to use nifedipine to block CAV1.1 or the HPR and see what happens when we stimulate the fibers in this way. As CAV1.1 is upstream the action of ATP, is upstream the P2Y receptors, we should expect that no change will be produced by inhibition of a protein that is upstream the action of ATP. So we performed the experiment, but instead of expected, we found that transcriptional events induced by ATP stimulation were lost when fibers were pre-incubated with a CAV 1.1 blocker. So this experiment told us that the transcriptional events that occur after ATP stimulation are dependent also on CAV 1.1 activation. So we come back to our results that showing that ATP induced a small depolarization to understand what were happening there. So we took fibers in a standard solution containing 5 millimolar external potassium, and we construct a calibration curve of membrane potential versus different concentration of extracellular potassium. In blue, you can see the values of membrane potential we obtain for different concentration of extracellular potassium. In red, we can observe the points given by the Nernst equation to different extracellular potassium concentration, and we found that they are not so different. So it's a good calibration curve. So we wanted to find the way to have fibers depolarized at the point they reach when are stimulated with ATP. So that is minus 50 millivolt to minus 40 millivolts. So this could happen if we place the fibers in an external medium containing 20 millimolar of external potassium. In this manner, we should have fibers at a resting potential of minus 40 millivolts. So in this form, we work with 20 millimolar external potassium as a stimulation, and we first measure calcium signals when we stimulate electrically at 20 Hz and when we add 20 millimolar external potassium. So in the empty squares, you can see the typical calcium signal we obtain when we stimulate the fibers at 20 Hz with the high elevation of calcium that is the product of ryanodine receptors opening, causing the contraction, and this slow decay that has been related to the opening of IP3 receptors. So when we made the experiment of changing the fibers from a normal potassium external concentration to 20 millimolar extracellular potassium, we have a slow rise in calcium levels that reach also slow values of calcium. But more interestingly, when we inhibit IP3 receptors, we observe that we lost the small decay that we have already published, and more important, we lost the calcium signal that we were able to see after stimulation with 20 millimolar external potassium. So according to the previous results, when fibers are at 20 millimolar external potassium, an increase in IP3 intracellular levels was observed compared with the previous situation when fibers are at the standard Ringer solution. And importantly, we also see the transcriptional change that we have already observed when we stimulate the fibers at 20 Hz. So in this graph, you can see that mRNA levels of slow isoform of troponin I are increased, and the fast isoform is decreased. In the same way we have when we stimulate the fibers at 20 Hz. In both cases, this process is blocked when an inhibitor of IP3 receptor is used. So we want to remember you our ATP release, to coming back to the fact that this ATP is coming out in two peaks. The first one between 15 seconds and 1 minute after the electrical stimulation, and the second between 3 to 5 minutes after electrical stimulation. So here the two other question appears. Why the two peaks, and why it's the role of each one? Is the same, is not the same? So for test that, we have the difficult task to separate the appearance of the two peaks to be able to analyze them. So until now we have not found the way to have the second peak without the first peak, okay? But we did find a way to eliminate the second peak and maintaining the first peak. So this way was to perform an electrical stimulation at 20 Hz, observing in control fibers the typical ATP release, but in the experimental condition, we add apirase to hydrolyze ATP, and in this way, 1 minute after electrical stimulation, we were able to eliminate the second peak of ATP, because probably they are hydrolyzed by apirase. But more importantly, when we saw the transcriptional change after this experiment, we observed the expected change obtained with electrical stimulation, but these changes were lost when the second peaks were no longer present. So with this experiment, we can affirm that ATP release in its second peak is needed for gene expression, or saying in another way, the second peak of ATP is the transcriptionally active. So we knew that the activation of signaling pathway that finished with IP3 production at diacylglycerol production will also activate a series of PKC activity. So we looked for the second peak in the presence of a PKC inhibitor as BIM, and we found that this second piece was greatly inhibited in the presence of PKC inhibition. To try to explain that, I show you a schematic picture from Cosoli et al., where they show that when they activate G protein couplet receptors that will produce IP3 and diacylglycerols, there will be an activation of PKC that will be able to phosphorylate chloride channels, in this case CLC1, that is the most abundant chloride channel in the muscle cells, and this phosphorylation will be closed the channels, reducing the chloride conductance. So to test this hypothesis that a chloride conductance will be implicated in the depolarization and the transcriptional events we have seen, we performed an experiment when fibers were electrically stimulated in the presence of normal ringer with a nominal chloride content, and in the presence of a reduced chloride ringer solution. So we observe the classical ATP curve when normal ringer is used after electrical stimulation, but we can see also that the first peak is unaffected by the use of low chloride ringer, but the second peak is greatly inhibited by this change. So implying that flux of chloride ions are needed to the developing of the second peak. So, well, to summarize all of these results, we come back to our cartoon. So we will still have the polarization at 20 Hz that will be sensed by CAV1.1, activation of pan-exit channels, allowing the exit of ATP that will act on purinergic receptors to activate a signaling cascade that will end with IP3 production and diacylglycerols. This diacylglycerol could activate PKC that in turn could phosphorylate CLC1 that will produce a small depolarization of the fiber that is sufficient to reactivate CAV1.1 without production of muscle contraction, but enough to reactivate the signaling cascade that will in turn produce more IP3 that will then bind to its receptors to cause calcium signals dependent on IP3 receptors that will in turn activate transcriptional change related to muscle plasticity from fast to slow phenotype. I thank you for your attention and I will be glad to answer to your questions.